Today we will study the pathology of cervix, and first of all we will study the neoplasia of cervix. To study the neoplasia of cervix, let's have an idea of the anatomy and development of cervix. This region is known as cervix, and cervix is composed of two parts. One is endocervix and the outer one is exocervix. So before telling you about the neoplasia of cervix, let's first discuss that how this cervix develops. So first of all, we'll study the development of cervix. That this endocervix is lined by columnar epithelium, whereas exocervix is lined by squamous stratified epithelium. The endocervix is lined by columnar epithelium and the exocervix is lined by squamous stratified epithelium. During the puberty, there is this columnar epithelium, this squamocolumnar junction. It extrudes outward. During puberty, this squamocolumnar junction, it extrudes outward. So the new structure will be somewhat like this. And this region will be having columnar epithelium. And there will be the old squamous epithelium.
This is the part which developed on the Mullerian tract.
So what happened during puberty? During puberty, there was extrusion of squamocolumnar junction so that the columnar epithelium is exposed outside into the exocervix. But now this region will undergo metaplasia. This region will undergo metaplasia, and that metaplasia will be squamous metaplasia, so that the new structure will be somewhat like this. This was our mature old squamous epithelium. There is stratified squamous epithelium. And now this columnar epithelium will undergo squamous metaplasia. But there will be a problem. There will be a big problem that this squamous epithelium will now be known as immature squamous epithelium. This epithelium will be immature squamous epithelium. This epithelium is mature squamous epithelium, and this epithelium is immature squamous epithelium because it is formed by the metaplasia of the columnar epithelium. And here is our old columnar epithelium in the endocervix. Got this point? So, but here, this region is now dangerous. This region is now dangerous because HPV virus can infect this region, and this region is known as transformation zone. So we just discussed the formation of transformation zone. We just discussed the formation of transformation zone. And HPV virus can infect HPV virus, human papilloma virus can infect this transformation zone. So till now we have discussed that HPV virus is having tropism for this immature squamous epithelium. Now let's say, and now what are the risk factors for occurring in HPV infections? Do you know any?
Multiple sex partners.
Multiple sex partners. Multiple sex partners, or having a male partner who is having other previous sex partners, or having a male partner who is having other previous female sex partners, and early age of first intercourse. If these risk factors are present in a person, so she's more prone to acquire an HPV infection. Now, if someone acquires an HPV infection, let's say we will now magnify this region. Let's say these are those immature squamous cells of the transformation zone. Immature squamous cells. And HPV virus. HPV virus is a DNA virus or an RNA virus? HPV virus is a DNA virus or RNA virus?
DNA.
HPV virus is a DNA virus. So this will infect these squamous cells. When these squamous cells will be infected, HPV virus will use the host cell DNA polymerase. HPV virus will use the host cell DNA polymerase. Let's just magnify it now. This is one squamous cell, and this is HPV DNA. It will use the host cell DNA polymerase to replicate. But there is a problem that as this cell replicates, as the DNA polymerase of this original cell, host cell, is working, so it will also replicate. And when the cell replicates, it matures, it stops further replication. Is it clear? When this host cell matures, it stops further replication. And why is this so? Why is this so? This is because of two very important genes, RB genes and P53 gene. This cell is having breaks. This cell is having DNA breaks, and these breaks are RB and P53. When this squamous cell matures, these RB and P53 will act as brakes, and they will stop further replication of these cells. And when the further replication of these cells stop, our infection, our HPV infection cannot spread. The HPV cannot spread. But HPV is quite clever. HPV is quite clever. It has overcome this issue by having two proteins. What are those two proteins? Now, these proteins are E6 and E7 respectively. Okay. First of all, I have to specify that this HPV is a high risk HPV. This HPV is a high risk HPV. Okay. Now it has infected. This HPV has infected our squamous cell, and its DNA. It uses the DNA polymerase of the host cell, and it replicates It replicates and it produces two very important proteins, and these proteins are E6 and E7. It produces two very important proteins that are E6 and E7, and E6 will knock out RB, and E7 will knock out P53. E6 will knock out RB and E7 will knock out P53. And now that braking mechanism is lost, that braking, the tumor suppressing mechanism is lost, RB and P53 will not be working, and this infection will spread. And when this cell will keep on proliferating without any breaks, and this cell will keep on proliferating, and this is the start of our lesion. Okay, but there is another thing. That is very important for the tumor progression, and that is only present in high-risk HPV virus. So first of all, to start to have an idea of high-risk HPV virus, we will first differentiate high-risk HPV virus from low-risk HPV virus. How to differentiate it? There are some major differences. A low-risk HPV virus produces these two proteins. A low-risk HPV virus produces these two proteins, but these two proteins are somewhat weaker and different than that of high-risk HPV virus. A low-risk HPV virus and high-risk HPV virus. What is the difference between a low-risk HPV virus and a high-risk HPV virus? These two E6 and E7 both are produced by a low-risk HPV virus as well as a high-risk HPV virus, but here they are weaker, and here these E6 and E7 are very stronger. It is the first difference. What is the second difference? A low-risk HPV virus will always cause an episomal infection. Whereas high-risk HPV virus will cause integration. It integrates into host cell DNA. And this is very important for the tumor progression. Why? Low-risk HPV virus cannot integrate into the host cell DNA, whereas a high-risk HPV virus can integrate into the host cell DNA. And that is the reason a low-risk HPV virus will cause a condyloma only, whereas high-risk HPV virus can cause squamous intraepithelial lesions and then invasive squamous cell carcinoma of cervix. Why? Because it integrates into the host cell DNA. And now you will be asking a very genuine question that why is this integration very helpful? You will be asking a question that what's the reason behind this? And I will tell you reason just now. That let's say this is our host cell and this is host cell DNA. And a high-risk HPV virus, the high-risk HPV virus integrates into the host cell DNA. There will be two things happening now. Most of the time, this high-risk HPV virus integrates into the host cell DNA near some proto-oncogenes. Most of the time, this high-risk DNA integrates near proto-oncogenes. Let's say there are proto-oncogenes. And this integration near these proto-oncogenes accelerates them and they convert into
Oncogenes.
So this is very important into the tumor progression. Secondly, this HPV virus DNA will be having some will have these E6 and E7 proteins, but normally the genes for E6 and E7 are not working. Normally they are suppressed. Normally there is a suppressor. Let's say this is a gene A, and this is a gene for E6, and this is a gene for E7. This A is normally suppressing E6 and E7, but due to integration, this suppressor will itself be inhibited, and E6 and E7 will be free to transcribe and result in the formation of many E6 and E7 proteins respectively. So what was this concept? And this concept was that integration is very important. Integration into the host cell DNA is very crucial for the progression of a tumor. And why is this so? This is because of two reasons. Number one is that when a high-risk HPV gene, high-risk HPV DNA, it integrates near some proto-oncogenes. These proto-oncogenes will be converted into oncogenes, and this oncogenes will be responsible for the tumor progression. Secondly, there are normally some inhibitors. There are normally some inhibitors of E6 and E7 that are present in the HPV DNA. So after this integration, this response is lost. This inhibitor is lost, and E6 and E7 will be free to transcribe, and we will have a lot of E6 and E7 protein that will knock out RB and P53 respectively, and the infection will spread. Now, the HPV infection we have studied till now, but HPV infection alone cannot cause the tumor progression to invasive squamous cell carcinoma. HPV alone cannot cause invasive squamous cell carcinoma. It needs some other factors to be also be present. And number one of that factor is there can be some immunodeficiency. And this immunodeficiency, what is the most common type of immunodeficiency in HPV infected patients? HIV infection. HIV infection, because both are transmitted sexually. Both these infections are transmitted sexually, so most of the time, if a person is having an HPV infection, he or she can also have an HIV infection. And this HIV infection will cause immunodeficiency in a patient, and this immunodeficiency is a plus point for the development of the invasive squamous cell carcinoma. Why? This tells us a very interesting thing. This tells us that our immune system plays a role in the suppression of HPV. Our immune system plays a role in the suppression of HPV. So if the immune system is not working properly, the HPV will progress. The second thing. The first thing was immune deficiency, and the second thing is some hormonal deficiencies. And the third is some other sexually transmitted diseases. These three factors favors the progression of the lesions caused by HPV infection. We'll study the course of HPV infection in the second video. Thank you.